Hello everybody. Let me thank the organizers for inviting me to give this talk about the FunCoup database of networks and show you some tools that we developed to analyze these networks. So I'm going to quickly go over the contents of the database and then show you then some ways that you can use this to get biological insights with some unique tools that we have for our database, such as comparative interactomics and for network-based pathway analysis, and also some tools for finding disease genes and disease modules. And finally, I'll show some benchmarking also that came out recently. So here's a paper where you can find all the details. I'm not going to show many details during this talk, but they're all in there. So FunCoup is a functional association network database, and what we mean by that is both interactions. So it's all about interactions between proteins. So it could be a physical interaction, but it could also be an indirect interaction. So genes that influence each other functionally. And those are combined, you could say, to get a very large network. But then you can see also what type of interaction that's stored in the database. So it's built and it's constructed with a method called weighted naive Bayesian integration here. And essentially what we do is we have positive gold standards, sort of a reference data set, and negative gold standards. And with those, we can use different types of evidences and see how much they are, how much information they have relative to these gold standards. And that gives us, in the end, 5 different networks for each species that can either be used separately or merged into one. And there's also extensive orthology transfer, so the evidence could be in a different species. And these 10 evidences that we have right now are, so it's either direct protein interaction or it's correlations, for instance, correlated gene expression, correlated regulation, or correlated evolution, for instance, as this phylogenetic profiles. So right now we're focusing on species that have a lot of data, and that gives us 22 species in total, mostly eukaryotes, but also some bacteria and archaea. And during the pandemic, we also added SARS-CoV-2 as a host virus genome interactome. And I'm not going to really go into the details, but just point out that if you look at the statistics, you can see that most of the evidence comes from actually correlated gene expression. So that's sort of the largest contributor. And if you look at what species the evidence came from, then you can also see that it's mostly actually, so that's in panel E here, it's mostly coming from other species actually via orthology transfer. There are some exceptions, so some species are more based on their own data, but there is extensive transfer of information between species. So what do we get and what does it look like? Well, you can take a look at the human network, for instance, with this plot from the Graphister tool, where you can see that it's actually a very dense network. It has many very big hubs, and then some structure in there, but it's actually very, very dense. So this is about 5 million links then for 18,000 proteins. All right, so let's show them some of these tools. So the front page, the landing page looks like this. You can type in your gene identifiers in the box here, or you can, there's also a button we added called SARS-CoV-2 search. And what that gives you is this host virus network, where the blue nodes are the human genes and the red ones are the viral genes. So there are no interactions between the viral genes because that's not in the data. But you can use this in a couple of different ways. So for instance, if you look at the spike protein, which is known to be the viral protein that sort of attacks the human cell, it's known to bind to these 4 genes. And so for instance, but then you can also go one step beyond that using the human network. So you can see, well, what happens after, or what binds the thing that spike binds. So for instance, we found this SLC16A1 gene that later turned out to be quite related to COVID. So it gives you interesting candidates for how the viral infection proceeds. Okay, so another thing that was added in this release was regulatory links. So with the direction from transcription factors to their targets. This is quite early stuff. It's a very small fraction of the links that actually have a direction. And we're working on improving that for the next release. And there's also added like if you're a biologist and you want to study the network in a particular tissue, then this is, for instance, the network that comes out of querying FunCoup with this gene set, thyroid hypoplasia. But some of these genes are not expressed in the thyroid, so they're not that relevant actually for this particular query. So, but you can filter it, for instance, for the thyroid gland genes. And then you get out only the subnetwork that's relevant in that tissue. Okay, so those are the new features. Here's an old feature, actually, but it's unique to FunCoup as far as we know. And you can, what you can do is you can submit a query in one species, but then ask it to align so networks in other species using ortholog links. So here we have a query in human. Those are the blue nodes, and C. elegans in red. And normal solid lines are interactions, and these horizontal green dashed lines are ortholog connections. So here's an example. For instance, we wanted to study Alzheimer, like how well is that conserved in C. elegans. So a few Alzheimer genes were input, presenilin and FLNB. And then you get, well, a number of orthologs in C. elegans, and you can see some of those are connected to. And in particular, if you look at this sort of square configuration here, where you have two, I should probably turn on this mouse pointer. Well, never mind. Where you have PSEN1 and cell 12, the orthologs, and they interact with TMED2 and cell 9. So indicating that that part of the pathway is conserved between human and C. elegans. And TMED2 is not really a known Alzheimer gene, but probably should be studied and can be well studied in C. elegans. So that's an example of how to apply this compared to interactomics. Another example is just to see, well, what's the level of conservation? If you put in some glycolysis genes, you can see that in human and mouse, as expected, it's super conserved. Whereas, so that's blue and orange, and the red parts are, let's say, the subtilis, where there were actually no interactions conserved there. Okay, so let me move on to how to use FunCoup for pathway enrichment analysis. Now, a little background of this is that So what we want to do is you did your experiment, you have your genes, and you want to find out what pathways are then enriched for your query set. There are, in general, 4 categories of methods: classical overrepresentation, and more advanced methods, including network enrichment, where you look at more the enrichment of links in between your query set and the pathway. And recently, there was a benchmark paper published by in my group, and also by Davide, who is here in the audience, so you can talk to him more about that, which was actually the first benchmark comparing all these 4 different approaches with each other. So on the network crosstalk enrichment, we developed another method, not going to talk about that, which has a very nice null model that behaves well because the pathways are kept intact, so there's no strange bias going on, which some methods have a problem with. And there's an online tool for this Anubix method that's called PathWix. So if you're in the FunCoup on the website and go to the interactions tab, you can link directly then from the subnetwork that you have to this PathWix, which has prepared. So it's using 3 different pathway databases and can analyze pathway enrichment for you. And then now going away slightly from FunCoup website to the PathWix website, which looks like this, where you get well, the list of pathways that are enriched, the significance of that, and also a matrix of how many connections there are between the query gene and the pathway. You can also see that in a network configuration. So, but let's get back to FunCoup and use it for finding disease genes. So there's an algorithm that we call MaxLink, where let's say we have some disease genes, in this case only 3. We can map into the network, we can find the neighbors, and then we can also do some statistics on that type of geometric test to see, well, which ones have which of the new genes that were not in the disease gene set are significantly connected to the disease gene set. So that's called MaxLink, and on the website there's a tab called MaxLink. You can put in your seed genes, and you get out a network like this. So for instance, this is an example of myasthenia gravis with 3 known genes. And you can see, so in the circle, that's the query set. And outside the circle are then the found new candidates and their connections to this disease genes. You can also get a list with the p-values from that. So that's a way to find sort of the most significantly linked new candidates. We also have another approach called Topaz, which has a slightly different goal. It's also finding disease modules, but it's trying to take, let's say, all known disease genes and connect them to each other and trying to find a single module out of that. So it's a top-down approach that basically starts with a fully connected full seed network and then removes links. Either keeping only sort of single step connections or 2 or 3 step connections. So in this case, for instance, it took if you took a look at took all the disease genes for biliary liver cirrhosis, there were 16 separated islands that could be connected into one single module. But that's not yet in this current release online, but it will be in the next one. So let me spend 1 minute on... 5 minutes. Yeah, okay. There was a recent paper, so now you might be asking, well, how good is FunCoup relative to other databases? And I acknowledge that there are many other of them. So there was a paper where they looked through some 40 odd interactomes and benchmarked them. So recent paper that's only on bioRxiv so far by IDECR group. And well, in that benchmark, FunCoup came in 3rd place, so that's nice. Of course, there are so many different ways of doing benchmarking. Here they used BizDNet in the GWAS catalog as references, but there's always a chance of bias, like what if some of these were trained on that? So we did it ourselves too. And took the database OrphanNet with the idea that those are rare genes that aren't for which there isn't that much data. And then we actually, this is in our own hands, we found that FunCoup outperformed the HumanNet and the string networks. Okay, so by the way, I should mention that there are some online tutorials on YouTube. I didn't really show you how to do it hands-on, but these videos do actually show you how to do it hands-on. So you can either just find a link in the help page, or go to the search for the Sonnhammer Lab channel. So to sum up this then, it's one of the largest databases that's out there that has good benchmark performance. We have this comparative interactomics tool and directed links as unique features, plus also now the tissue subselection. Plus, then I mentioned some of these extra tools then, like for pathway analysis, disease gene detection, and module analysis. I should point out that there is a new release in the making, and there's a poster by Davide on the 262, which shows you some preliminary results on that. And the new additions there is actually the whole website is improved. There's also a Cytoscape plugin and an API, and full integration of tools, some of which are not yet fully integrated, and also improvements in comparative interactomics and regulatory links. And actually, this contains 640 species networks with a particular one. And it's all done with a new type of Bayesian, bin-free Bayesian training. So with that, I would like to acknowledge people that have created the database: Emma, Miguel, David, and Dimitri. And thank you for your attention.
Erik, that was a beautiful presentation, and it's wonderful to see the evolution of these fundamental tools that the community has been relying on for some time. But it raises, of course, new questions and potential features. When you did that very elegant comparison between species, there were two fundamental concepts that I think you used. One, the species intra-species relationships between the genes, and then the comparison of the networks, which I thought was elegantly placed out. I don't know if that's done automatically. That's beautiful.
No, you have to do it a bit manually. Yeah, for sure.
So I wish. But the question I have is, have you thought about combining what you have, what your tissue specificity, what your species specificity? These are built on canonical backgrounds. But you've already showed that you can mask that per tissue. The question is, can you do a case versus control sort of environment where you have a diseased individual or a diseased model and a pure model or a true model that's controlled? One has the canonical background, but the other one has the disease state condition. I wonder if you thought about that or how you're considering that sort of that in your development.
Okay, thanks. Very nice question, Win. And the answer would have to be that it's not something that is available right now, and I guess we have maybe thought a bit about it, but it's not really, yeah, it's not that obvious how to do it, I would say. But maybe we can talk, we can brainstorm a bit later. It's a good idea, I think. Yeah, very nice.